All right, so this last video for cellular respiration, we're going to look at the last three must-knows. We're going to look at how pyruvate is moved from the cytosol, the cytoplasm, into the mitochondria and introduced to the citric acid cycle, how electrons from NADH and FADH2 are passed to a series of electron acceptors to produce ATP by chemiosmosis, and the roles of the mitochondrial membrane, the proton gradient, and ATP synthase in generating ATP. I know it's a long video, but don't be afraid. Just take it all in slowly if you can and appreciate the beauty and the complexity of these processes. All right, so we're going to start by just reviewing where we left off in the last video. In the last video, we started off by looking at this molecule right here called glucose. Glucose is a sugar molecule, and originally this molecule came from photosynthesis. So photosynthesis built this beautiful molecule. It took some carbon dioxide from the air and some water from the roots, and it rearranged those atoms to build this molecule of glucose. And if we look closely at glucose, we can see that it's made up of 6 carbon atoms in the center. So 1, 2, 3, 4, 5, 6. 6 oxygen atoms, 1, 2, 3, 4, 5, 6, and 12 hydrogen atoms, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. So the chemical formula for glucose is C6, 6 carbons, H12, 12 hydrogens, O6, 6 oxygens. And in this molecule, we've got a ton of chemical energy. Chemical energy is stored in the bonds of a molecule, and the energy in this molecule originally came from the sun, but the plant captured that energy and stored it in the bonds of this molecule. So this molecule has so much energy, and what your cell is going to be doing in cellular respiration is this. It's going to start breaking apart this molecule and using the energy that's originally stored in the bonds of this molecule to power all the processes that happen in your cell. So we're essentially trying to harvest, we're trying to pull out energy from glucose. Remember, think of glucose as like a sponge, which is full of water, and you can squeeze out that water, and that water can be used for other things. In the same way, this molecule of glucose is full of energy, and you can release that energy to be used in other places. So remember, whenever you're breaking down a molecule, you're catabolizing that molecule, you're breaking it down, there's a release of energy. It's an exergonic reaction. And exergonic reactions, when you release energy, can be used to build up new molecules. And so that's really what we're doing here. We're taking this really high power molecule, high energy molecule, and releasing the energy to be used other places. Now, the energy eventually will be stored in a smaller molecule called ATP, adenosine triphosphate. And think of this glucose molecule kind of like a $100 bill. It's got so much chemical energy, it's so valuable, but we're going to be slowly breaking down this molecule and releasing energy that's eventually going to be stored in smaller molecules called ATP, but multiple of them that can be spent around the cell like dollar bills. Okay, so the whole point of cellular respiration is to take your glucose, to chop it up, and to get energy from it. Okay, now that's just like a very broad overview. In this video, we're going to be breaking it down into a lot of steps. There's going to be a lot of words, there's going to be a lot of numbers, and it can be a little bit overwhelming, but I want you to always come back to this. We're chopping up glucose to get energy for our cell. That's the main point. Okay, so let's start by talking about adding some vocabulary and reviewing from our last video. So we know glucose looks like this. Glucose will go into our bodies when we eat food, and it will travel through our bloodstream until it eventually finds a cell. The cell needs energy. So the glucose will come in, it will be shuttled in through a protein channel here in the cell membrane, and it will go into your cytosol, your cytoplasm. And this glucose molecule, this 6-carbon molecule, will be cut up in a process called glycolysis, cutting glucose. And what's happening here is you're going to take the 6-carbon sugar and you're going to cut it into 2 smaller pieces, 2 3-carbon sugars. And those 3-carbon sugars are called pyruvate or pyruvic acid. Okay, now what you're actually doing here as you're cutting this up is you're also removing some hydrogens, right? And whenever you're removing hydrogens, we call that oxidation, right? Oxidation, oil rig. Oxidation is loss, oil. Oxidation is loss. So you're losing hydrogens. And remember, hydrogens carry with them electrons. So this is a proton and an electron. That's a hydrogen atom. And so you're actually removing some electrons. And think of electrons as kind of like energy, right? So if you remove electrons here, you're removing energy from this molecule. And those electrons or those hydrogens can be can be attached to something new and it will pick up the energy. It's basically carrying the energy. So let's look here. What molecule picks up those electrons or those hydrogens that were originally stored in glucose? Well, it's this NAD+. Notice that this NAD has a plus sign. The plus sign means it's positively charged. And so it's going to, if it sees a negatively charged particle around, it's going to latch onto it. So there are electrons floating around, and they are going to be combining with hydrogens, right? So simple proton, so just one proton plus an electron makes a hydrogen. And so when those come together, they're going to bind onto this NAD+. It's going to grab those electrons and hydrogens, and it's going to create NADH, right? So this NADH has been reduced. It's gained an electron, right? It's become more negative, right? It was originally more positive. Now it's been reduced. It's gained an electron or gained a hydrogen is another way of thinking about it. And those hydrogens came from glucose, right? And we know when this NADH has gained that electron has energy in it. So now this NADH is full of energy and it can be shuttled around and moved in other places. Okay, think of this NADH as an energy carrying molecule that can move around the cell, and it can pass this electron or this hydrogen to other molecules, right? This can be broken down again into NAD+, it can be oxidized, and that can supply energy. Okay, so think of these hydrogens as like electrons, and these electrons have energy. Whenever you're thinking about moving electrons, you're thinking about energies being passed. Now, in this process, you're also going to generate a little bit of ATP, 2 ATP molecules from breaking down glucose. Okay, now you have this pyruvate molecule. If you have oxygen present, this pyruvate will go into the mitochondria, and basically you're going to continue to chop it up. You're going to continue to oxidize your molecule. You're going to chop up those hydrogens, remove those electrons, and pass them other places. And eventually, your whole goal is going to be to make some ATP. These are the energy currencies for the cell. This can be spent for multiple places around the cell as energy. Okay, so remember, the whole point is to get from glucose to ATP, and there's a bunch of steps in between. The whole thing we're doing is we're chopping up glucose, we're oxidizing it, we're moving electrons, passing those electrons other places.
Attention lines. Attend today's BSU
Thank you,
meeting
Obi.
in room 101.
Okay, so if there's no oxygen present, or if you don't have a mitochondria, right, if you're a prokaryotic cell, then you will move into fermentation. And why do we do fermentation? Let's review from the last video. Well, notice here, if you have a bunch of reduced NADH, right, this NADH will build up. And so you actually need to get it back to NAD plus so it can be reduced again, so it can accept those electrons from glucose. And so what you're going to do is you're going to do something called fermentation. And in our cells, we do something called lactic acid fermentation. But in like bacteria or yeast, they do something called alcoholic fermentation, which produces down here a molecule called ethanol instead of lactate, which is how you make alcohol. But in our cells, if we don't have enough oxygen, like if we're running a race and we are breathing really heavily, we're using up all our oxygen, we run out of it, we go into fermentation, we're going to break down pyruvate into lactate. And remember, this is can be toxic to our cells. It changes the pH, which makes our muscle cells feel very sore, right? When you're running really fast without enough oxygen. But basically, the whole point of this is to you are trying to oxidize your NADH back to NAD plus to be replenished. That's why you do this whole thing. All right, so just to review, that process here is anaerobic. It doesn't require oxygen, and all cells can do this. But if you have oxygen present, you can move back. You can actually move this pyruvate into the mitochondria, and you can break it down even more to get even more energy out of it, right? This molecule isn't done. It's got more hydrogens, it's got more carbons, it hasn't been fully broken down, it hasn't been fully oxidized yet. All right, so let's kind of summarize this. Glycolysis, we take a glucose, we take a glucose molecule and we invest some ADP and some inorganic phosphate, and we also invest some NAD+, right? And that glucose is going to be chopped up into 2-pyruvate, and overall we're going to be creating 2 ATP, and this is so important, 2 NADH molecules, okay? These 2 NADH molecules now have that hydrogen. This is going to shuttle energy around for other reactions that are happening later. So we talked about what happens. You're going to run out of NADH eventually, or you're going to run out of NAD plus eventually. So how do you recycle your NADH back to NAD plus? You do fermentation, like we talked about. Okay, we just talked about this. So fermentation happens when there's no oxygen and you're recycling your NAD+. All right, now once we have our pyruvate, remember there's two pyruvates, two 3-carbon molecules. If you don't have oxygen, you go into fermentation. If you do have oxygen, now this is what we're going to get into next. You go into the next step called the Krebs cycle or the citric acid cycle, which is going to happen inside the organelle called the mitochondria. Okay, just to recap, glycolysis, we're inputting a molecule of glucose, we're inputting some NAD+, we're inputting, it requires some ATP, and we're inputting some ADP and inorganic phosphate. What are we getting out of it? We're getting 2-pyruvate, we're getting 2 reduced NADH molecules. Now this has the energy from glucose. We get some ADP, and then we create 4 ATP total, but we gain 2 because we invested 2 molecules of ATP. So all that math, it can get kind of complicated, but just know that glycolysis produces 2 ATP total. And we'll skip this. Okay. So I want to talk a little bit about the evolutionary perspective, like thinking about how this connects to the evolution of life on Earth. So there was a time when there was life on Earth without any oxygen, without free oxygen, O2 in the atmosphere. So you think of like bacteria, right? The original prokaryotes. Energy had to be captured from organic molecules in the absence of O2. So bacteria also eats glucose, it eats sugar, but doesn't have mitochondria, first of all. And there was a time where there was no oxygen to be able to do next steps aerobically. So organisms evolved glycolysis, and these are the ancestors to all life. This happened before endosymbiosis, and all organisms still use glycolysis to get some ATP from food. But the problem with this, why don't we all just stick with glycolysis, is remember, you don't get that much ATP from one glucose molecule. You oxidize your glucose, you create some ATP, but that molecule can still be broken down even more. You just need energy. You just need some oxygen for that. But that's okay. For 1 billion years, life on Earth survived with just glycolysis. So these prokaryotes survived. But the only problem is you're not getting all the energy out of glucose that you can. Think of it like taking your sponge and only like taking like a tiny bit of water out of it. There's still so much left in the sponge, right? So that's the downside. So what that resulted in is slow growth and so slow cell reproduction. So the prokaryotes grow more slowly and reproduce more slowly than they could if they could harvest all the energy from glucose. Some other notes I have on this, just so you know, you don't have to memorize this, but scientists predict that bacteria evolved on planet Earth 3.5 billion years ago. Okay, 3.5 billion years ago, bacteria evolved, and they used glycolysis to make ATP from glucose. And then around 2.7 billion years ago, bacteria evolved the ability to photosynthesize. Okay, so they evolved the ability to photosynthesize, and this happened 2.7 billion years ago. The product of photosynthesis, we'll get into this later, is some oxygen. So now you're creating an environment 2.7 billion years ago with some oxygen in the atmosphere. Combine that with the evolution of eukaryotic cells. So about 1.5 billion years ago, we think that the first eukaryotic cells came about through endosymbiosis, right? So you have this cyanobacteria. So this bacteria is able to do photosynthesis, right? It's able to capture energy from the sun and produce O2, oxygen. So now you got an oxygen-rich environment on the planet Earth. Then this bacteria is engulfed by another prokaryotic cell, creating a eukaryotic cell, right? And now this becomes your chloroplast. Your chloroplast now can do photosynthesis inside a protected area. And then we learned also about how eukaryotic cells evolved mitochondria as well by engulfing another prokaryotic cell. And that mitochondria now can do cellular respiration in the presence of oxygen. So just some kind of history of the planet and how life evolved along with changes and how life got energy basically from glucose and also using oxygen now. So glucose is only the start, or glycolysis is only the start of this whole thing. So you start off with your glucose, you make 2 molecules of pyruvate, but there's more carbons here that you can't oxidize. So these carbons have more hydrogens attached. You can get more energy out of it. So let's continue on to looking at the next step after glycolysis, what happens next? Let's just do a quick little overview and then we'll get into each one. So we just finished glycolysis, glucose to 2-pyruvate. You're creating some NADH, energy-carrying molecules that have electrons. These NADH molecules are going to be used later at the end. So hold on to this. Remember, these NADHs are carrying energy, and you're producing a little bit of ATP. This is happening in the cytoplasm of your cell. Then this pyruvate will now be shuttled into your mitochondria, okay? In the presence of oxygen, you're going to do 3 more steps. The first is called pyruvate oxidation. This is also called like the transition phase or the link phase from your POGL. And then we're going to get into the citric acid cycle, also called the Krebs cycle. You're going to be creating more ATP, and then you're going to be creating more reduced molecules, these high energy carrying molecules. And then the last step, oxidative phosphorylation, also called the electron transport chain, also called chemiosmosis, is going to be producing basically the most ATP. We really are trying to get here. The whole point of this whole thing is to take your glucose and to generate ATP. And these steps are going to get us there. Okay, so let's take a look inside our mitochondria. So remember, we just made our pyruvate molecules. They're going to be shuttled into our mitochondria, but I want us to kind of look at the structure of the mitochondria. And this might be something you want to kind of draw a diagram in your notebook or write down, just so you can kind of have these words familiar in your mind when we go over this more in detail. So here's our mitochondria. Notice that it's double membrane. There's a double membrane. And the reason for this is because the endosymbiotic theory thinks that perhaps once upon a time, right, this mitochondria was its own prokaryotic cell, and it was engulfed by another cell, right? And so this inner membrane was the prokaryot's own membrane. This outer membrane came from the membrane of the cell that engulfed it, right? So as the prokaryotic cell, as the mitochondria originally came in, it got wrapped around, kind of like packaged up by the membrane of the engulfing cell, now has a double membrane, okay? Now, outer membrane, inner membrane. We have this space, which is really important for you to know about, called the intermembrane space. Inter means between. So the intermembrane space is the space between outer membrane and the inner membrane, okay? Now, inside the mitochondria, if you're to like bite down into it, the stuff inside is called the mitochondrial matrix. So the matrix is the space inside, right? Here we have in blue, this is a picture taken with an electron microscope. The blue is the intermembrane space. We also have these little folds here. They're called cristae. This is the location of, eventually we're going to talk about the electron transport chain. So that happens in the folds here, the cristae, okay? And we talked about the matrix. I think that's it. Now remember, why are there folds in the mitochondria? Think back to when we learned about membranes and surface area. And remember that the more folds that you have, you've increased the surface of mitochondria. And so basically, a lot of chemical reactions can happen in the folds here of the mitochondria because there's so much surface area. It's kind of the anatomy of the mitochondria. It's the powerhouse of the cell. And in this next part of the video, we're going to be learning about what happens inside and what happens in this inner membrane space. Okay, so let's take a look at what happened where we left off. So we have our pyruvate. Our pyruvate is going to be shuttled into the mitochondria. And now we're in the mitochondria. We're living in here. We're in the matrix. Now we're in the space in between. And we are going to do a process called pyruvate oxidation. Okay, pyruvate oxidation is also called the transition stage or the link stage, like your POGL says. Or it's also called the preparatory reaction. Lots of words here. But here's what's happening. Remember how we took our 6-carbon glucose and we chopped it up in half? We made a pyruvate molecule. Pyruvate has 3 carbons. So if we look here, we have 1, 2, 3 carbons. Okay? And there's 2 of them. So in total, you still have 6 carbons. And in this reaction, what you're going to do is this. You are going to take this carbon here and this oxygen, and you're going to chop it off. Okay, you're gonna hydrolyze this. You're gonna chop that bond. What would happen if you chop this bond? What do you have left? You have CO2 and then hydrogen. Notice here that what we're making is carbon dioxide, CO2. Okay, that came from our pyruvate. But there's no hydrogen. Where'd the hydrogen go? Well, this hydrogen is now going to be stripped off, right? It's going to be oxidized. The pyruvate is going to be oxidized. The hydrogen is going to be stripped off. And where is it going to go? Remember, hydrogen's full of electrons. It's got energy. You can't waste that. It's not just going to disappear. The hydrogens now are going to attach on to the NAD+, and the NAD+ is now going to be reduced. It's gaining electron. Reduction is gain. So now we have NADH here. Okay, we've reduced. And now this molecule is carrying energy, right? Because it has gained an electron or a hydrogen atom. Okay. Now, what happened to the pyruvate? Well, remember, there was two of them. There was two pyruvates, right? So we created two total NADHs because two hydrogens were stripped off from two separate pyruvates. Now, what happens to the rest of this pyruvate, the two carbons that are left over? Well, those two carbons and this oxygen and these hydrogens, they're going to attach on to something called CoA, coenzyme A. And this CoA now is attached to these two carbons, and now we call it acetyl-CoA. Okay, so acetyl-CoA is these two carbons, this oxygen from the original pyruvate with a CoA. And why are there two of them? Well, remember, we had two pyruvates. We make two carbon dioxides because we had two of these, right? Two pyruvates are going to create two carbon dioxide molecules. So what have we done? We've done more oxidation. We have removed more hydrogen from our original molecule. That's the whole point. We're trying to break apart this molecule, remove electrons, and pass them on somewhere else. So now this NADH is going to be, we're going to come back to it. Hold on to this thought. It's carrying the energy, right? We're tracing the energy. We're tracing how the atoms are moving. Okay, here's another picture of it. So here's our cytosol. So we have our 2-pyruvate that came from glycolysis. We have a 3-carbon sugar. And remember, there's two of them. So we still have our 6 carbons from glucose. We're going to transport it into the mitochondria, past the mitochondrial membrane, and it's going to go into the matrix. You strip off the CO2, right, and you're creating carbon dioxide, and that's what we're breathing out. So the carbon dioxide that you're breathing out right now likely came from this reaction happening in one of your cells. At the same time, the hydrogens right here are going to be reducing NAD+, NADH, and now that's carrying energy, right? Now, the leftover pieces of the pyruvate combine with coenzyme A to create acetyl-CoA, which is now your new product. So this pyruvate oxidation, what does that mean? What is pyruvate oxidation? You're taking a pyruvate, you're oxidizing it. Remember, oxidation is loss. It's losing energy. It's losing electrons, right? You're trying to take this original molecule and you're trying to get energy shuttled somewhere else. So we're moving energy, we're moving electrons, we're moving hydrogen, and we're oxidizing our pyruvate oxidation. Okay, so again, our CO2 has been oxidized, right? This is an exergonic reaction. Whenever you're pulling something off, you're pulling off those hydrogens, you're releasing energy, right? The hydrogens are carrying energy. You're releasing energy. It's exergonic. And exergonic reactions are used to create or reduce or create basically new molecules. Endergonic reactions, building up, anabolism. And you need that to build up this new molecule of NADH, right? NAD plus is pretty stable. Adding on an electron is going to require energy. It needs energy to do this, to build a new molecule, to add a piece. Think about if you're building Legos, it takes energy to add on another piece to your Legos. So as you're reducing NAD plus to NADH, as you're adding that hydrogen, you are using the energy from the oxidation of CO2. And what do you have left? So now you have a 2-carbon compound. So acetyl-CoA now has 2 carbons, and you've released some CO2. That's going to be released as waste, and you've made some NADH, some energy-carrying molecule. Okay, so then let's look, and remember, this happens twice. There's 2 acetyl-CoAs, right? Because we started off with 2 pyruvates. All right, so another way of looking at this, here is our pyruvate. This came from glycolysis. Here's a cytosol. Here's your pyruvate. Remember, there's 2 of them. And this is just a basic model. There's actually carbons and hydrogens attached, but this is a basic model. It's shuttled into the mitochondria, into the matrix, pyruvate oxidation. You're removing a carbon dioxide. And the hydrogen also goes to this NADH, NAD plus to be reduced. Coenzyme A attaches to the 2 carbons. Now you have acetyl-CoA. Remember, you have 2 of these. We've oxidized pyruvate. So here's a summary slide. If it helps you to see it in words, here are some words. We've just finished glycolysis, and now we know a little bit more about pyruvate oxidation, or the transition stage. this step, we're oxidizing our molecule even more, we're creating even more NADH, and we're eventually going to get to the payoff stage where we're going to be making tons of ATP. All right, so let's get to the last two steps of cellular respiration. We've gone through glycolysis, we've gone through pyruvate oxidation, we are oxidizing our original glucose molecule, and we are producing a little bit of ATP. But really, a lot of the energy so far has been going to these reduced energy carrying molecules, or NADH. And you're going to notice here, just a little preview, we're next going to get into something called the citric acid cycle or the Krebs cycle. And notice here that we are going to take our acetyl-CoA that we just made, right? Remember, our acetyl-CoA looks like this. It has 2 carbons and it's attached to this coenzyme A. And we are going to oxidize it even more. We're going to take off even more hydrogens and we're going to create a little bit of ATP. But really, the whole point here is going to be to pass some of those hydrogens or electrons or energy to our energy carrying molecules, NADH, and a new one called FADH2, which will eventually combine with the reduced energy carrying molecules, NADH here, in this last step called oxidative phosphorylation or the electron transport chain or chemiosmosis, where we're really going to get most of our ATP. Okay, so let's take a look at where we've been. So we just talked about, again here, we got our pyruvate molecule, our 2 pyruvates, these 3 carbon molecules that originally came from glucose, right? They're going to go into the matrix of the mitochondria, and we are going to oxidize this pyruvate molecule 2 times, right? Because there's 2 of these. We're going to create some NADH, and we're going to create acetyl-CoA with giving off some carbon dioxide, right? So now we have this acetyl-CoA, and it's going to be broken down even more. So there's going to be a series of reactions here. It's called a cycle, a series of different steps breaking down this acetyl-CoA even more. And notice here that we're going to be producing some carbon dioxide. We're going to be reducing some NADH. We're going to make a little bit of ATP. We're going to make another energy carrying molecule called FADH2 and even more NADH. So really the whole point of this is to squeeze out as much energy as we can from this acetyl-CoA, and we are going to be trying to create these energy carrying molecules. That's the whole point. Okay, so let's start by looking at what's happening to this original acetyl-CoA as it's going into our citric acid cycle. So remember, we left off with our pyruvate in glycolysis is a 3-carbon molecule. Remember that 2 pyruvates are made in glycolysis. And then through pyruvate oxidation, we have resulted in 2 molecules of acetyl-CoA, and each of those molecules has 2 carbons in it. So let's pay attention to what happens to the carbon skeleton of this original molecule. Remember, these carbons are originally in glucose. What happens to them as this sugar molecule continues to get oxidized? We call it a sugar because it's this carbon-based molecule. So what we're going to do is we're going to take our acetyl-CoA, and we'll just take a look at one of these for now. And remember, it's going to happen twice because we have 2 of these acetyl-CoA molecules. And we are going to add a 4-carbon sugar to our acetyl-CoA. So basically, we're going to build something new. Okay, we're going to attach 2 pieces together, 2 molecules together, and we're going to create 4 plus 2 is a 6-carbon molecule called citrate. There's a lot of names to this, there's a lot of numbers, but just kind of allow yourself to kind of see basically what the whole point of this is. How are we oxidizing this original molecule to get energy out of it? Okay, so we're going to take our citrate, and we are going to take off a carbon and 2 oxygens. Okay, so imagine we were to take this and break off a carbon and 2 oxygens. We're going to produce some carbon dioxide. Okay, so now we've lost a carbon. So now our original skeleton, right, now has 5 carbons. Then we are going to be ripping off another carbon, okay, and another 2 oxygens. And we are going to be now having a 4-carbon sugar. Then we are going to keep going. Okay, so basically we end up with a 4-carbon sugar. That 4-carbon sugar can now then be recycled again. If there's another acetyl-CoA molecule, it can attach onto this and it can go around again. Okay, now remember this happens twice. So if 1 glucose molecule creates 2 pyruvates, 2 acetyl-CoA, and so this happens twice. So we're oxidizing sugar. Now, as all of this is happening, we are also reducing these molecules as well. we are reducing some energy carrying molecules, excuse me. Okay, so we are actually oxidizing these carbon skeletons. We're going to pull off some hydrogens from these and pass them to our energy carrying molecules, to our NADH and our FADH2. Okay, so let's look at our electron carriers now. So once again, we have our pyruvate, 3 carbons. We have oxidized it. We've created some NADH in the pyruvate oxidation stage, and we have now taken off a CO2. If you think back to our pyruvate oxidation, we have our acetyl-CoA, 2 of these. We add on our 4 carbon to make a 6 carbon sugar, citrate. Okay, we already talked about that. We already also just talked about how we pull off a carbon dioxide, so now we have a 5 carbon sugar. But at the same time that you're doing that, you're also going to be oxidizing this carbon sugar here, and you're going to be pulling off a hydrogen or an electron or energy, and you're going to be reducing NAD plus to NADH. I didn't write the NAD plus here, but you remember that NAD plus gets reduced to NADH. It's gaining that electron or that hydrogen. And this is now carrying the energy that was originally stored in the bonds of this 6 carbon molecule. Okay. Then we're going to do it again. We're going to pull off the carbon dioxide, and we're also going to pull off a hydrogen. We're going to pass it to NADH. NADH is now the Postmates carrier. It's shuttling around that hydrogen, and we're going to use that later to make some ATP. At this phase, we actually make 1 molecule of ATP. This is mixed up here. My apologies. This should say, I'll fix it here. This should say ATP. I mixed it up. And this should say ADP. Oops, sorry about that. So we're going to phosphorylate ADP. We're going to add on a phosphate, and we're going to make ATP in this step. Okay, the details of that you don't need to know. Just know that 1 molecule of ATP is made here. Then, let's see if I can get back. Okay. Then you are going to now reduce your 4 carbon molecule again. So you're not removing any more carbons, but you're pulling off another hydrogen, and you're pulling off 2 hydrogens actually. You're taking your FAD2 plus molecule, and you're adding 2 electrons and 2 hydrogens to reduce FADH2. And so now this is going to be carrying some energy, right? It's gained some hydrogens. It's been oxidized. Oh, no, sorry. It's been reduced. My bad. It's gaining hydrogens here. And then you're going to create another NADH. You're going to reduce another one. And this happens again because remember, we have two acetyl-CoAs. So in total, we're going to make 1, 2, 3 NADHs the first time, and then it goes around again. So 6 total NADHs in the citric acid cycle. The first time around, we make 1 FADH2, but then remember, it goes around again for our second acetyl-CoA, and now we're making 2 FADHs. So even though we only got, because this goes twice, we only got 2 ATP total from the reduction, or the oxidation, of these carbons. So why do we spend all this time oxidizing this carbon molecule? Well, really the point is so that we can pass those hydrogens or those electrons to these energy carrying molecules. All right. So thinking back to our original glucose molecule, C6H12O6, we've now fully oxidized our glucose. So we've now ended up with a bunch of carbon dioxide. Carbon dioxide has no hydrogens, right? It's been fully oxidized. It's lost all its hydrogens. And in total so far, we've only gained 4 ATP. Think about how go back all the way over here. Okay. In glycolysis, we produce 2 ATP net. In the citric acid cycle, when it goes twice around, we produce 2 ATP. So total, we've only gotten 4 ATP, but we've fully oxidized our glucose. We've taken off all the hydrogens we can, and we've gotten as much energy as we can out of it. But where is the energy now? Even though there's not as much ATP, the energy is hiding, right, in these intermediate electron carriers, in these NADH and FADH2 molecules. They are now going to be paying off eventually and helping us to make our ATP. All right, so going back here. Okay, so we've gained a net of 4 ATP, but our last step, we are going to be taking these energy carrying molecules, these NADH and FADH2 molecules, and producing ATP. Here's another picture of the Krebs cycle, looking at like the details of how the carbons are shuffling around, changing. But remember, really what you need to know here is that the point of the Krebs cycle is to make these energy-carrying molecules. So once again, the whole point, the Krebs cycle produces large quantities of electron carriers, NADH and FADH2. They're stored energy, right? They've gained electrons. Electrons are energy, or they've gained hydrogens. So what's so important about these NADH and FADH2s? They are going to be generating energy later. Okay, so let's kind of look back. This is kind of another summary. We've gone through this already, but it's another way of thinking about it. So remember, after glycolysis, we ended up with 2 pyruvates, or pyruvate is a 3-carbon sugar. We've taken some NAD and FAD, and we have reduced it. We've passed these hydrogens from the pyruvate to these molecules, and now they've picked up some energy. Now, in total, after we take our pyruvate, we do pyruvate oxidation, we do the Krebs cycle. Now we end up with 4 NADH and 1 FADH2 from this whole process. And if we just did 1 pyruvate. And then we get 1 ATP from this, and we end up with 3 carbon dioxide molecules. Okay, if you were to look back at our Krebs cycle here, you'd end up with 2 carbon dioxide molecules from the Krebs cycle. And then don't forget, back in pyruvate oxidation, back here, we created a carbon dioxide too. So 1 carbon dioxide and then 2 here, right? So that's for 1 pyruvate. 3 total carbon dioxide molecules from this whole process, right? And this happens twice. So in total, we've gained 6 carbon dioxide molecules, and those are what we breathe out. We're looking at our ATP, a net gain of 2 ATP from this step here. So we've gained 2 ATPs from pyruvate to carbon dioxide, plus the 2 ATPs that we got from glycolysis. And once again, the pyruvate, there's 2 of them. This happens twice. So we end up with 8 NADHs and 2 FADH2s. Okay, so we're using a lot of numbers here. We're using a lot of terms. The most important thing for you to really understand is the whole point, which is we are trying to pass energy to these energy-carrying molecules. The details of all the numbers are less important, but just helpful for you to kind of understand where everything's moving around. Okay, so again, why the Krebs cycle? If we only make 2 ATP from the Krebs cycle, we only made 2 ATP from glycolysis. What's the point? Well, remember, those NADHs and FADH2s are so valuable. They're electron carriers. They're reduced molecules. They've gained electrons, which store energy. And eventually they're going to be used in the electron transport chain. So we're slowly releasing chemical energy from the bonds of glucose. It's not just one quick release of energy. Through many, 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 many steps, we are slowly releasing this energy and basically building up as much energy as we can. If you think back to earlier in this lecture, we looked at these pictures, right? If you were to compare the breakdown of glucose, the oxidation of glucose, to a hydroelectric system where water is flowing through a turbine, if you took all the energy here and you allowed it to flow down and generate some transfer energy here, basically, you're gonna, once all the water's down here, it's lost all its energy, it's transferred all its energy here. And that's gonna give you a little bit of energy for a time. But if you break it up into multiple steps, you can actually harness even more energy from this reaction, from these multiple reactions. So here, you're reaching something called equilibrium faster, right? The water is basically transferring all its energy elsewhere faster, whereas this takes more steps to gain equilibrium. All right, so that's kind of why we spend so much time breaking down glucose. Okay, I think that's it for now. And the whole point is to make ATP. All right, so then we're going to move on now to the last piece. This is our final step. We've made it, the electron transport chain. Okay, so what is the electron transport chain? We finished glycolysis, we finished pyruvate oxidation, we finished a Krebs cycle, we've made a little bit of ATP, right? We've made 4 ATP total so far, but we've really made a ton of energy-carrying molecules, NADH and FADH2, and we're going to now cache those in to generate a bunch of ATP using something called the electron transport chain. So we're in this step here. We're in the mitochondria, and it's going to be actually important for us to know a little bit more about the structure of the mitochondria. so we can kind of know where we're at for this process. So remember, the mitochondria has a double membrane, outer and inner. The inner membrane is folded, right? It has a high surface area, and there's a fluid space in between the two membranes. It's called the intermembrane space. This is going to be really important for you to know. The intermembrane space. So it's like between this and this. Then the mitochondrial matrix is a space in between, like in the inside, basically the inside of the mitochondria, and it's filled up with fluid. So for this last step, the electron transport chain, we're going to be in the intermembrane space and the matrix here. So where we're at so far, just to recap, we've made some ATP in glycolysis. We haven't made any ATP in oxidation of pyruvate. Remember, we're basically just taking our sugar, we're oxidizing it a bunch, we're pulling off those hydrogens. And we made some ATP from the Krebs cycle, but we need to get some more energy in this last step. Okay. So those NADHs and FADH2s from glycolysis, the 2 that are made in glycolysis, the 8 NADHs and the 2 FADH2s from pyruvate oxidation, the Krebs cycle, will be now cached in. And in order for me to kind of explain this, I'm going to show you this video here, and it's going to help you kind of visualize what is happening in the electron transport chain. So what is happening here? Well, that's sort of fast. Okay, so where are we? We are actually in the mitochondrial matrix. So the mitochondrial matrix is going to be down here, and up here is going to be the intermembrane space. So we are, the top of that part is right here in the intermembrane space. The bottom is in the mitochondrial matrix. And there's a membrane, right? So there's a membrane between, right here, we're right here in the inner membrane, between the intermembrane space and the matrix. And it's still a phospholipid bilayer, right? It's made up of a bunch of lipids. And this is separating that intermembrane space from the inside the mitochondrial matrix here. And notice here, embedded within this membrane, right, we have a bunch of these protein molecules. And actually, if we were to zoom in, we'd actually see like smaller proteins actually combine together to form something called a protein complex. So these are protein complexes, but just to simplify it, they just show you one piece here to simplify it. So there's a bunch of these proteins. And here, we're going to watch what happens as, actually, let me see if I can slow it down. Slow this down here. As our electron carrying molecules, our NADHs and FADH2s are now going to be oxidized. They're going to lose their hydrogens and they're going to pass their energy on somewhere else. So if we start off here, that's going to go really slow, with our NADHs. Now, our NADHs have 2 electrons that they're going to be now releasing. Now, with those electrons go some hydrogens too. So it's going to release these electrons. It's going to lose a hydrogen. Okay. Now, we are going to watch as the electrons are transported in the electron transport chain. Okay. So those 2 electrons came from the NAD and the H. Both lost an electron, and you've lost the hydrogen as well, right? That NADH has been oxidized into NAD+, and it's lost 2 electrons. Think about your POGL that we did in class. Now, these electrons, think of them again as energy. These electrons are going to supply energy to do a very special job. Their job is they are going to pump these protons, these hydrogen ions, into the intermembrane space. Okay, this is the intermembrane space. Here's the mitochondrial matrix. Okay, so we've pumped them out. And then what have we done? We passed on the electrons, the energy, right, to this next molecule here. Boom. Oh, now we take our next energy carrying molecule, FADH2, which has electrons. We donate those electrons, and we lose 2 hydrogens, right? It's becoming oxidized. FAD now becomes oxidized into FAD2+. Now we've lost those hydrogens, and those hydrogens are now going to be shuttled against their concentration gradient into the intermembrane space using the help, the energy of these electrons. So the whole point is for these electrons to help move these hydrogens out. Then they're going to be passed along here. It's going to be shuttling these electrons or transported to the next protein complex here. And we are going to get shuttle even more hydrogens out until our final step here, we are going to get our oxygen is now going to accept these final electrons. They're going to be final electron acceptors. These electrons need to go somewhere. They can't just hang out forever. So oxygen is going to pick up these electrons. And at the same time, there's 4 electrons here. They're going to gain, each oxygen is going to gain 2 hydrogens. So the hydrogen's coming with an electron. Now that's going to allow this hydrogen to bond here with an electron. 2 electrons, 2 electrons. And now you've created H2O, 2 of them, 2 H2O molecules. And that's going to be a waste product of cellular respiration. Now that you have, notice here, a high concentration of hydrogen ions in the intermembrane space here and a low concentration here. Now, what happens if you have a high concentration of an ion on one side of a membrane? Well, now you notice here that there's a higher positive charge here. It's more positive on the outside than the inside of the matrix. And so what you have here is an electrical gradient. An electrical gradient is when there is a difference in charge or a voltage. You can even talk about a voltage is a difference in charge from one side to the other side of the membrane. Okay, so we also see that there is a difference in concentration in hydrogen on the outside versus the inside, right? So there's a higher concentration of hydrogen here, and we know this is a chemical gradient. Chemical gradient means there's a higher concentration on one side. This total is called an electrochemical gradient because there's going to be a strong pull for these hydrogens to get back into the mitochondrial matrix. So the diffusion, the facilitated diffusion, we're going to use a protein to help these hydrogen ions pass back in down their concentration gradient, right? No, it doesn't require any energy, right? We're trying to make energy. So we're going to use this process, kind of like river flowing through a dam, right? In order to generate some energy here. So notice here that our hydrogens are now going to go and be shuttled through this enzyme called ATP synthase. Synthesis means to make. We're trying to make ATP. And our hydrogen ions are going to be diffusing down their concentration gradient. And this process is going to supply energy. It's going to supply energy here. We got this phosphate group that binds on here. And now we call this oxidative phosphorylation. We are now phosphorylating or adding another third phosphate group. Remember, that takes energy to build ATP. We're now building ATP using an ADP molecule and an inorganic phosphate. And this ATP synthase is providing the energy to do that. So if you notice here, now you've created ATP. And this happens again and again and again. All right, I think that we covered all that. Now, the last thing I want to show you here, though, is what would happen if, so you had your whole electron transport chain, but let's say you had a buildup of electrons at the end and there was no final electron acceptor. You had no oxygen, right? Let's say you stopped breathing. Well, the electrons would have nowhere to go. They couldn't be accepted. And this would basically stop the whole process. So you wouldn't be able to pump any more hydrogen out, and you would shut down your ATP synthase and you would make no ATP. And this is why you would die. And this is how some poisons work. They shut down this process here so that you can't actually have oxygen be the final oxygen acceptor or electron acceptor. And now ATP synthase cannot do its job. All right, so I think I got it here. All right, I think that's it. So I'm going to post this video on your Unit 3 guide too. That way you can kind of watch it over again if you need. All right, so we kind of went over broadly what's going on. I know it's pretty complicated, but let's look at it again. So where are we? Here's the membrane. Here is the intermembrane space. Here's the mitochondrial matrix. And we're going to start here at the top of the electron transport chain. We take our NADHs, we are going to oxidize it. So we're going to strip off that hydrogen and pump it out using the electrons that also come with that hydrogen, right? We saw that in the video. So this is a protein complex of electron carriers. It's going to shuttle this electron. It's going to pass this electron to this next piece here. We're going to oxidize FADH2 to FAD. These electrons now are going to be passed here. Now, as these electrons are passing, they're providing the energy to shuttle hydrogen ions out. Those hydrogen ions came from the oxidizing the FADH2. Okay, and then you're going to keep passing the electrons here and then here, and finally, oxygen will be accepting those last electrons along with some hydrogens. The hydrogens plus two electrons are now going to be here reducing our oxygen. So we're reducing our oxygen, we're adding some hydrogens, and now we make water. So the whole point of this was to generate a concentration gradient of hydrogen. We're going to use this enzyme here called ATP synthase to allow facilitated diffusion of hydrogen ions down. This is going to turn this like turbine here. Well, I'll show you a picture later of ATP synthase, which will then do something called oxidative phosphorylation, which basically is adding on this last phosphate to ADP. That takes energy, and we are going to generate ATP. Another word for this is chemiosmosis. So chemiosmosis is diffusing hydrogen down its concentration gradient and allowing ATP to be created through this, right? So this whole thing is called oxidative phosphorylation. So that's how we're making ATP here. All right, so once again, if you look here, we are in our intermitochondrial membrane, or inner mitochondrial membrane, and we're pumping the hydrogen ions into the mitochondrial matrix like we just saw. Now, a way that we sometimes talk about the electron transport chain is that the electrons, remember the electrons are being pulled off of this NADH, we're oxidizing it, and we are passing electrons from protein complex to protein complex to protein complex, right? And we talk about these electrons as flowing downhill. So basically, as the electrons are flowing, they're being passed on to a new protein carrier or protein complex that basically lowers the energy of that electron. So at the beginning, the electron's like really excited, it has a lot of energy. And as it goes down, it's kind of like rolling down a hill. It's losing energy as it goes until finally it's going to be accepted by this last oxygen. Okay, here's another picture so you can kind of see where this is taking place. It's important for you to know the inner membrane space versus the mitochondrial matrix is the direction where hydrogen ions are moving in. Okay, here's another way to see it. So the electrons, as they're moving in steps from each carrier, they're moving towards more electronegative molecules. So those electronegative molecules are basically molecules that are really selfish for electrons. So as you go, so like this molecule right here is more electronegative than this one right here. So it's basically drawing the electrons towards itself. That's why the electrons are able to be passed, because there's a new, think of it as like a magnet. It's pulling, it's selfish for electrons. It's pulling electrons towards itself. So those electrons are like, okay, fine, we'll go. And then they keep going on and on and on until we reach the most electronegative, which is oxygen. Oxygen is very electronegative. So it's going to suck those electrons towards itself and finally accept them. And this is controlled. It's controlled oxidation because we're slowly pulling that electron. It's kind of like a game of hot potato. One of these electron carriers is taking it and passing it on to the next. And as we're doing that, we're kind of releasing energy piece by piece. And remember that energy is being used to shuttle those hydrogen ions out into the intermembrane space. All right, until eventually that electron is now accepted by oxygen, and those electrons go along with some hydrogen ions to make H2O. Okay, so I think we kind of covered this. If you want it in words, you want to pause this and look at it again, you can take a look. Okay, so we talked about this, but just to go over it again, why did we build up all these hydrogen ions in the intermembrane space? Because we have this enzyme, ATP synthase, that has the ability to allow these hydrogens to pass through. And as they're passing through, this thing called the cylindrical rotor is going to spin. So there's literally this thing that spins and it moves these hydrogen ions in. And as it does, it's like this like little knob here. It's like this is like a little machine in your cells. It basically cranks out these ADP plus inorganic phosphate to make ATP. We talked about how hydrogen's flowing down its concentration gradient, and basically I think we covered all that. We're making a bunch of ATP. Okay, now something to note, because you might see this come up in questions or just something to know is this process right here is called oxidative phosphorylation. It's a special type of making ATP. However, all the other steps so far of cellular respiration, we're using a different type of phosphorylation called substrate level phosphorylation. Okay, here's basically what it means. So, for example, when we were back in glycolysis in the Krebs cycle, whenever we were making ATP, we were taking ADP and making ATP. So, like for example, right here, remember in the Krebs cycle, we made some ADP plus phosphate to make ATP. When we were doing that, we weren't using ATP synthase. Instead, we were actually taking an enzyme and we were bonding as substrates this inorganic phosphate and this ADP molecule. And basically, this enzyme was changing shape once these substrate bind and it's producing a product. It's releasing that phosphorus or that phosphate group, it's attaching it onto the ATP, it's providing energy for this to work, and then it's releasing whatever was carrying that phosphate. So, substrate level phosphorylation is when ATP is made using an enzyme. Where oxidative phosphorylation is this. You're using this ATP synthase, which is basically spinning here, allowing these hydrogen ions to flow down, and it's producing ATP. Here's a picture of the protein if you were to see it using a computer model. This is just a protein molecule. It's an enzyme, and it's a beautiful molecule because this is the reason why we're alive. We're making all this ATP in using ATP synthase. We talked about chemiosmosis. So we're coupling the electron transport chain. We're using this buildup of hydrogen ions so that they can flow down and produce ATP. So this is the better way of making ATP. This is the payoff phase. We have these molecules in the mitochondrial membrane that are these integral transport proteins. So like we saw, these transport protein complexes that are shuttling the electrons. They are transport proteins embedded in the membranes between the inner membrane and the mitochondrial matrix. And yeah, basically, we're yielding about 32 ATP molecules from one glucose. So scientists kind of debate how much ATP is made, but I've heard anywhere from 28 to 32 ATP molecules from one glucose in the electron transport chain. So that's much more from the two from glycolysis and the two from the Krebs cycle. But the only problem is this only occurs when there is a presence of oxygen. So this is in aerobic respiration. Why is that? Well, remember, oxygen is our final electron acceptor here at the end of the electron transport chain. And so this it requires oxygen to happen. All right, I think we're almost there. Okay, so now we're gonna do some summary just to kind of put it all together because we've talked a lot and I know there's a lot of words, a lot of numbers, but remember the whole point is to take glucose and to make ATP. And the way we do that is we chop up glucose to 2-pyruvate. We are going to reduce NAD to NADH, two of them. We make 2 ATP. We move the pyruvate into the mitochondria matrix and we oxidize pyruvate again. We basically pull off some hydrogens and we add them to NADH. We're making electron carrying molecules and we make acetyl-CoA. The acetyl-CoA goes into citric acid cycle and produces some ATP and then recycles that original carbon skeleton to go again and again. And we're producing some more energy carrying molecules. And then finally, the energy carrying molecules, the NADH, the FADH2 from all these steps are now going to be passing their electrons to the electron transport chain, which will shuttle those hydrogens into the intermembrane space here, which will then diffuse back into the mitochondrial matrix through chemiosmosis or oxidative phosphorylation, which generates 26 to 28 ATP. So for each glucose molecule, we can make a total of about 30 or 32 ATP molecules. Now, some of the energy from the original glucose molecule is actually released off as heat too. So you could have made more ATP if energy was completely conserved in this reaction, but some energy is released as heat according to the second law of thermodynamics. Anytime you're transferring energy between different forms, some energy is released off as heat. Okay, this really puts it all together. I know it looks like a lot, but we've broken it down. So let's review. But glucose to 2-pyruvate, making some ATP, reducing NADH. NADH will then eventually go to the electron transport chain. We'll come back to that, right? It's carrying its 2 electrons and its hydrogens, electron carrier. The pyruvate will be shuttled through this protein into the mitochondria. Here's our outer membrane, here's our inner membrane space, here's our inner membrane, and here's the mitochondrial matrix. Here's our cristae, here's the folds here, and you can see how it's increasing the surface area. And that's important because now you can do a bunch of this all over the mitochondria. There's a bunch of ATP synthesis because you have so much surface area of the cristae here. This is basically putting everything we've learned this whole year together, which is so cool. Okay, so now we have our pyruvate in here. We're going to chop it up even more. We're going to pull off some carbon dioxide. We're going to reduce some NADH. The acetyl-CoA, this is pyruvate oxidation. The acetyl-CoA will, again, continue to oxidize your carbon skeleton, and you're going to pull off some more carbon, pull off carbon, make some ATP. You are going to reduce to FADH2, NADH. You're going to make 3 of those in this reaction. So 3 from the Krebs cycle. It's also called the TCA cycle. 1 FADH2 and 1 NADH from pyruvate oxidation. And now these are all going to combine here with the ones from glycolysis to the top of the electron transport chain. You've made some ATP here. Remember, this goes around twice, so we make 2 ATP in this step and 2 ATP in this step. Okay, so then we have our energy carrying molecules here, which are going to pass their electrons through the electron transport chain, which supplies the energy to pump hydrogen ions from the mitochondrial matrix into the intermembrane space, generating a high concentration of H plus ions. Also, a really positive charge compared to negative charge inside a voltage across the membrane. You are now going to be able to diffuse the hydrogen down its electron gradient, spin this turbine here, and now you're going to phosphorylate, oxidative phosphorylation, phosphorylate ADP to ATP. So adding on that last phosphate group, and now you're generating lots of ATP, and that ATP can be now used to do anything in the cell that requires energy. Now, not only glucose does this, so carbohydrates, but also you could take fat molecules and you could break them down also and produce acetyl-CoA. A fat molecule is really just a long chain of carbons and hydrogens. So you can actually get even more energy from a fat molecule, which is why we talk about fats as having more carbs than carbohydrate molecules, because fats basically have so many bonds, so many carbons and hydrogens, really a lot of high energy bonds. And you can oxidize that molecule. You can chop off those hydrogens and create a lot of NADH and FADH2s to eventually create more and more and more ATP. So this doesn't just happen with your sugars, it happens with the fat that you eat as well. All right, so we're almost there. So we took glucose, we added some oxygen, and we overall made 6 carbon dioxide, 6 waters, and around 36 ATP. Where did the glucose come from? It came from our food. Where did the oxygen come from? It came from our lungs, right? It was shuttled around in our bodies through our red blood cells. Where did the carbon dioxide come from? Remember, we were producing that in different steps throughout cellular respiration. One step was the pyruvate oxidation. We're making a CO2. The other step was the Krebs cycle or the citric acid cycle. We're making some CO2 as well as we are oxidizing our carbon skeleton that we started off with. Where did the H2O come from? Remember, we are passing electrons and hydrogens to oxygen, and we are reducing it as the final electron acceptor in the electron transport chain. The ATP came from a couple of different places. It came from substrate phosphorylation in glycolysis, in the citric acid cycle, and in the last step, chemiosmosis or the electron transport chain. What else is not listed? We did include our lovely electron carriers. We didn't include NADH and FADH2 because they're actually not involved in any of the changes in matter. They're just holding on to the energy for a little bit. So they don't really get recognition in this equation because they're really just the ones shuttling the energy around. Okay, and then why do we breathe? We need that oxygen to accept those electrons in the electron transport chain. So oxygen, we talked about this. If oxygen is unavailable, the electron transport chain cannot continue. We cannot pass electrons anywhere. ATP production ceases, we run out of energy, and we die. That's why we need oxygen to live. So cool, right? Okay, so the whole point is to make ATP. Now, we talked a little bit about how this can happen with fats too. So remember, a fat molecule is made from a glycerol and three fatty acid chains, three fatty acid tails, which is just a bunch of carbon and hydrogen bonded together. And those can do glycolysis. So you can actually chop those up and create some NADH. And then the skeleton will go into the citric acid cycle, and then it will go into oxidative phosphorylation. So the whole thing can still happen with fat. Even proteins. You can take protein amino acids and you can get energy from them by oxidizing the carbon skeletons that make up this biomolecule. Just to review really quick of fermentations, we talked about it in the last video, but remember if you have your glucose molecule that gets oxidized into pyruvate, but you don't have any oxygen present, we go into fermentation. If you do have oxygen present, you go into aerobic respiration in the mitochondria. And let's take a closer look at fermentation. So the product of fermentation, you could either make alcohol called ethanol, lactate or lactic acid, or some other products. So for example, let's say we were looking at a human muscle cell. Okay, so a human muscle cell, let's say you run out of oxygen and you reduce glucose to pyruvate, two pyruvate molecules, you make 2 ATP, but you don't have any oxygen. You are going to take that pyruvate and you are going to basically chop off a carbon dioxide molecule, CO2, and you are going to add on, you're going to pull off this hydrogen from NADH, right? You are going to oxidize it, you're going to pull it off, and you're going to try to regenerate NAD+. And that hydrogen is now going to go stick onto that this molecule over here. So it's going to add on that H here in place of this carbon dioxide, and it's going to be reduced. So this is going to pick up that hydrogen, and the whole point of this is so that you can regenerate NAD+. This will be converted into a molecule called ethanol. Okay, so if you see here, it's going to pick up even another hydrogen. So it's going to add a hydrogen here and a hydrogen here. It's going to pick up more hydrogens and produce ethanol, which is alcohol. You don't need to know all the biochemistry of this, but just see here that we're really just moving around some hydrogens so that we can regenerate NAD+, so that we can do glycolysis again, make more ATP. Lactic acid fermentation happens in some fungi and bacteria, and this is actually how we make cheese and yogurt. So the same process, similar process here, except we're actually, instead of ethanol, we're making something called lactate or lactic acid. And this happens in our cells. We make lactic acid in a muscle cell. So sorry, this is ethanol. This is not happening in our cells. We are making lactic acid, and this is what causes our muscles to burn when we run out of oxygen. Yeast and bacteria, this is how we make chocolate from cacao beans. So yeast is alive. It's a eukaryotic cell. Bacteria is prokaryotic cell, and they can do glycolysis. So they take the sugar molecules that are found in cacao, and they do fermentation and create chocolate. So this is how you make really good foods. Any fermented food happens from this process here, either producing ethanol or lactic acid. All right, so we've done it. We've finished cellular respiration. I know it was a lot. Use the extra videos that are supporting these videos on the You Must Know Unit 3 guide to help you. And good luck with your studying.